Um, when you say that there is no virus, I have I guess the context to understand what you're saying because I've dove into the papers that you reference often in discussing this. But for a lot of the people listening, what is it that you mean by that exactly? I know you touched on the theoretical portions of it, but I know some people will send me papers and be like, hey, they have done an intense genetic sequencing of the virus. What do you mean there's no virus? So what exactly do you mean by that?
So in order to say there's a thing, like a spoon, remember, we're not talking about a thought or a feeling, right, which you cannot isolate, but everything can be isolated. And if it's a fork, I can say this is an isolated fork and this is two isolated forks. So when the CDC says there are no quantified isolates of the coronavirus available, that means they've never seen the thing. And so they can't count it. Now, the question I think you're asking, so what are they seeing here? So the best way to describe this is to understand how human beings actually isolate anything. And if somebody wants to be my foil here, I think I can explain this so people understand it. Okay? So I'm going to ask you a series of questions, and then you try to give me your best answer. And just pay careful attention to the wording that I use, okay? So we're going to do an experiment to see if the caffeine in coffee beans causes high blood pressure, okay? That's our quest.
Okay.
So I take coffee beans, and I grind them up, and I put them in a capsule, and I give 10 people the ground up capsule coffee beans, and 10 people I give an appropriate placebo, and all the controls are appropriate, everything. Have I now proved, and they all get high blood pressure. None of the placebo, all 10 of the people I give the ground up coffee, all 10 of them get high blood pressure. Have I proven that caffeine in coffee beans cause high blood pressure?
No, you haven't, because you haven't isolated caffeine. You've done a coffee bean that's ground up and given it to the people.
Right. In other words, there's more things in there than just caffeine. There's fiber and caffeic acid and aromatic oils and maybe 100 other things.
Mm-hmm.
Okay, next step. I put some water in it, put it through a filter paper. I tell you in my study what the size of the holes in the filter is, and I end up with coffee, right? That's how you get coffee. I give that to 10 people. I give the other people the same water, so there's control. Have I now proven that the caffeine in coffee beans causes a high blood pressure?
No, you haven't.
Because
you still have coffee that is like being used in place of caffeine itself.
Right. In other words, there's more things than just caffeine. Right?
Yeah.
So, okay, I'm with you. So let's do a centrifuge, and it separates things by weight. I know the weight of the caffeine. It comes out in a band. I suck the band out. I do an independent chemical analysis, and it tells me there's only caffeine in this sample. I put it in a capsule. I give 10 people the same capsule, but it has nothing in it. And 10 people with the caffeine get high blood pressure. Have I now proven it?
I would say yes.
I would agree.
Okay.
Because we know the steps. We've isolated the caffeine. We can then characterize the caffeine. We can say how much it weighs and everything. And that is how you do science. And then you publish the independent chemical analysis so that anybody can see they got just caffeine. There's no fiber, there's no caffeic acid, there's no anything. And then usually somebody repeats that experiment and does the same thing and gets the same result. Got it?
Yeah.
So here's how from 1930 to 1954, they did the exact same thing with viruses. They took somebody with chickenpox or measles or something, and they took the secretions or the snot, and they ground it up like the coffee beans. right? Now, that's not the virus any more than that's the caffeine, right?
Right.
So they put it through a filter paper, and they got the liquid part where the virus supposedly lives, and they got rid of the cells and the fungus and the bacteria. And so now they had a bunch of, they had liquid, a supernatant they call it, and it has viruses and a bunch of other things. So then they did the next step, which is they centrifuged it in a gel electrophoresis. They pull out the molecular weight, and they show with an electron microscope that it's pure virus. Now, with measles, they couldn't find the virus. There was no virus. With chickenpox, there was. With herpes, there is. The next step, they give that purified, isolated virus. Then you can characterize the genome, right?
Mm-hmm.
You can sequence one end to the other. You can give it to an animal. None of the animals got sick.
Mm-hmm.
None of them. They could not, for 20 years, doing this properly, make any animal or person sick with a purified virus. And as I like to say, that was the end of virology. They were going to be plumbers then, except somebody said, there's another way to do this, which is what they do until today. They take snot, right? Respiratory secretions. They do a little bit of filtering. They take that filter stuff, like that's like the coffee. They inoculate that on a tissue culture, like monkey kidney cells. And lo and behold, nothing happens. It doesn't kill the kidney tissue. So then they figured out that if they starve the tissue, it's called minimal nutrient medium, that would help, but it still doesn't work. So then they figured that they had to put in antibiotics like gentamicin and amphotericin, which happen to be kidney toxins, and that then the tissue would break down and the tissue would release all these particles. And they said that means the virus grew and formed all these particles. That is how we prove to this day that the virus causes disease. Now, if you think about that, first of all, nobody pulled out the virus from the beginning. And just like you said, that the coffee doesn't prove it's the caffeine. And the second of all, we now know that when you poison any tissue, it defends itself by packaging up the genetic material. These are called exosomes or intracellular vesicles, and they detoxifies itself by secreting these vesicles, otherwise known as exosomes. In fact, if we stop the tissue from doing that, the tissue dies. Mm-hmm. So this is a fundamental way that the tissue detoxifies itself and then communicates through resonance to other organisms. Here's what happened. I've been poisoned by gentamicin. So every single one, and I mean this because people have sent me, literally hundreds of people have sent me title of the article, isolation of the coronavirus. Everyone is done this exact way. They take snot, and even in the Christian Drosten paper, they not only didn't have the virus, they were making a test for a virus. They had no examples of the virus, and they had no samples from any person with COVID-19. Now think about that. That's like, Tom, I want you to make a unicorn, a test for a unicorn, but you're not allowed to see a unicorn first. All you can see is a picture of a unicorn on the internet. Now, then you find a piece of a hoof and you say, this must be from a unicorn. Now, the fact of the matter is there could be a buffalo or a pig. I don't know if pigs have hooves. I think they do.
I think so.
It's zebra.
You don't have any idea whether it's coming from this so-called outside virus. And for people who don't believe me, here's a quote from magazine, July 2020, on extracellular vesicles and viruses. The quote is, "To date, there is no reliable method that guarantees a complete separation from viruses from exosomes or endocellular vesicles." And I can tell you there's a thing called a fork, and this is a different thing called a spoon. And the only reason you can't separate a fork from a fork or an exosome from a virus is because they're the same thing. All of these things we're calling viruses are excretions of the cell. And that's why in my first video I said you poison a cell and it poops out virus.